Assalamu alaikum everyone. In the course of hormones, our today's lecture is about eicosanoid hormones. What are eicosanoids? They are signaling molecules made by the enzymatic or non-enzymatic oxidation of a arachidonic acid, or we can say polyunsaturated fatty acids, which are similar to arachidonic acid. Arachidonic acid is a 20 carbon containing molecule, and eicosanoids are a subcategory of oxidized fatty acids of diverse carbon units in length and are distinguished from other oxylipins by their overwhelming importance as a cell signaling molecules. So eicosanoids are derived from 20 carbon containing polyunsaturated fatty acids commonly known as arachidonic acids. They have very short half-life, just about 10 seconds to 5 minutes. So it means that they are not stored, but they are produced at the site of action when they are needed. And functions are usually very limited to their actions on nearby cells. It means they are not transported from one cell to another. These eicosanoid hormones, they bind to specific cell receptor, G protein couple receptor, and generally increase cyclic AMP levels. They may also bind to nuclear receptors and alter gene transcription. Eicosanoids function in diverse physiological systems and pathological processes such as mounting or inhibiting inflammation, allergy, fever, and other immune response. They also regulate the abortion of pregnancy and perform normal childbirth. They contribute to the perception of pain, regulating cell growth. They control blood pressure and can modulate the regional flow of blood to tissues. In performing all these activities and roles, eicosanoids oftenly act as autocrine signaling agent to impact their cell of origin, or they may also act as paracrine signaling agent to impact cells in the proximity of their cells of origin, that is on their nearby cells. Eicosanoids may also act as endocrine agent to control the function of distant cells. So they are special cells which act as three modes of signaling agents. What are major classes of eicosanoids? There are prostaglandins, thromboxanes, and leukotrienes and prostacyclins. Mostly three classes. There are main three classes: prostaglandins, thromboxanes, and leukotrienes. And other are subclasses or subfamilies of eicosanoids. There are multiple subfamilies of eicosanoids, including mostly prominently the lipoxins and resolvins, and there is the sixth one, eoxins. So we can say there are six types of eicosanoids: prostaglandins, thromboxanes, leukotrienes, lipoxins, resolvins, and eosins. The first eicosanoid to be discovered were prostaglandins. They were first found in seminal fluid and were believed to be synthesized only in prostate gland. Hence, they were named prostaglandins due to the site of origin or location where they are produced in prostate glands. Prostaglandins were later found to have wide tissue distribution. They were found to produce physiological and biochemical effects on other organs too. There are some effects and functions of general eicosanoids: induction of inflammation, mediation of pain signals, induction of fever. They also act in smooth muscle contraction, including uterus during childbirth, smooth muscle relaxation, protection of stomach lining, simulation of platelet aggregation, inhibition of platelet aggregation, and sodium and water retention. There are the precursors of eicosanoids. As we have already discussed that eicosanoids are originated from a 20 carbon containing fatty acid known as arachidonic acid. The other precursors are eicosanoic acid, for example, alpha linoleic acid, which contains C18 carbon and is also known as omega-6. Arachidonic acid is also named as omega-6, while the third one precursor is eicosapentaenoic acid, which contains 5 double bonds and is named as and is categorized as omega-3. Here are structures for eicosatrienoic acid, which contain 3 double bonds, arachidonic acid, which contain 4 double bonds, and eicosapentaenoic acid, which contain 5 double bonds. Similarly, there is another thing very important to know, that when we talk about classes and families and subfamilies of eicosanoids, the types of eicosanoids depends upon the position and the location of double bond and the number of double bonds they possess. For each subfamily, such as lipoxygens, resolvins, and eoxygens, there is the potential to have at least 4 separate series of metabolites. 2 series derived from omega-6 fatty acids, and for example, arachidonic acid and linoleic acid. One series derived from, while the one series is derived from omega-3 fatty acids. And one series derived from even omega-9 fatty acid, which is not demonstrated here. This subfamily distinction is very important because mammals, including human, are unable to convert omega-6 into omega-3 fatty acids, polyunsaturated fatty acids. In consequence, tissue level of the omega-6 and omega-3 fatty acids and their corresponding eicosanoid metabolites, they link directly to the amount of dietary omega-6 versus omega-3 where they are consumed. It has often been suggested that the harmful results which are associated with the consumption of omega-6 fatty acid rich diets reflect excessive production and activities of omega-6 derived eicosanoids, while the beneficial effects associated with the consumption of omega-3 fatty acid rich diets reflect the importance and of omega-3 fatty acid derived eicosanoids. In this view, it means that we can say omega-3 fatty acids rich diets, they are much more beneficial as compared to omega-6 fatty acid diets. The opposing effects of omega-6 derived and omega-3 derived eicosanoids on key target cells or their final cells underlie the beneficial effects of these diets. This slide shows structural demonstration of prostaglandins. The first one, eicosanoids. As we can see here that starting from a prostanoic prostanoic acid, it's a 20 carbon containing molecule, and the difference lies, there are some other subfamilies or subclasses of prostaglandins. For example, prostaglandin A, prostaglandin D, E, F, G, H, and prostaglandin I. This classification is based on functional groups present at the position X and the position Y. It means that the functional group present at carbon number 9 and carbon number 11. There are also 2 other classes, PGF or prostaglandin PGF, which have 2 double bonds or which have 3 double bonds, and it depends upon the number of double bonds present in the linear hydrocarbon chain. There are structures of prostaglandin E1, E2 and E3, and if we look upon their structures, we can see just the difference lies in the number of double bonds on the linear carbon chains. PGE1 has just 1 double bond, PGE2 has 2 double bonds in his linear chain, and PGE3 has 3 double bonds in his linear carbon chain. While the structures of PGF1, PGF2 and PGF3 alpha shows again the presence and difference in the number of double bonds which are present in their linear side chains. While if we compare PGE1 with the PGF, PGE and PGF, we can see that the substitute which are present on X and Y position we have already discussed in last slides. Now I will present you some general introduction to prostaglandins. The first of all, it's very important to know how prostaglandins are different from other hormones which are produced by some glands of our bodies. We can say that unlike most hormones which are produced by glands and transported in the general bloodstream of our body and act on distant areas of the body, the prostaglandins, however, they are produced at the site where they are needed. Prostaglandins are produced in nearly all cells and are part of the body's way of dealing with injury and illness. Prostaglandins they act as signals to control several different processes depending on the part of the body in which they are made. They are made at sites of tissue damage or at the site of infection where they cause inflammation. They cause pain and the fever as part of the healing process. Pain and fever, they both of these signs, they are part of the healing process. When a blood vessel is injured, a prostaglandin called thromboxane stimulate the formation of a blood clot to try to heal the damage. It also causes the muscle in the blood vessel wall to contract. It means that it causes the blood vessel to narrow in order to try to prevent blood loss. While another prostaglandin, which is called prostacyclin and is very important class of eicosanoids, has the opposite effect to thromboxanes. It means that it reduces blood clotting and removing any clot that are no longer needed at the end of the bleeding. It also causes the muscle in the blood vessel wall to relax as compared to the function is opposed to the thromboxanes. Why? Because it want to dilate the blood vessels. The opposing effects that thromboxane and prostacyclin, both of these eicosanoids, they have on the width of blood vessels can control the amount of blood flow and regulate the response of the body's response or the tissue's response to injury and inflammation. Prostaglandins are also involved in regulation of the contraction and relaxation of the muscles in the gut and the airways. It means they are very important part of the respiratory system. Prostaglandins, they are known to regulate the female reproductive system also. So they are very important during pregnancy and childbirth also. They are involved in the control of ovulation, during menstrual cycle regulation, and in the induction of labor during childbirth. Indeed, manufactured forms of prostaglandin, that is the artificial prostaglandins. Most commonly, they are prostaglandin E2, which can be used to induce labor during our childbirth. How are prostaglandins controlled? Very important question to arise in our mind. The chemical reaction that makes the prostaglandins, it involves several steps. The first step is carried out by an enzyme which is known as cyclooxygenase. There are two main types of this enzyme, cyclooxygenase 1 and cyclooxygenase 2. When the body is functioning normally, baseline levels of prostaglandins are produced by the action of cyclooxygenase 1. While when the body is injured or any inflammation is occurring in the area of the body, the cyclooxygenase 2 is activated and produces extra prostaglandins, and it helps the body to respond to the injury. Then prostaglandins carry out their actions by acting on specific receptors. At least 8 different prostaglandin receptors they have been discovered so far. The presence of these receptors in different organs throughout the body allows the different actions of each prostaglandin to be carried out, depending on which receptor they interact with. So there are different receptors which are specific to the site of action and to the sort of function which has to be performed by the eicosanoids. Prostaglandins, they are very short-lived molecules and are broken down quickly by the body. The only they only carry out their actions in the immediate vicinity or locality where they are produced. This helps to regulate and limit their actions. What happens if prostaglandin levels are too high? High levels of prostaglandins, they are produced in response to injury or infection and cause inflammation. We have already studied that eicosanoids, as compared to hormones, they are produced just on the site of action and perform their function and are not stored. So, as far as any injury or infection or inflammation is any injury or infection occurred on the body due to some stimuli, prostaglandins are produced and cause inflammation. Inflammation is a sign to respond towards infections and injuries. They are associated with the symptoms of redness. Inflammation causes redness, it causes swelling, pain, and fever. So pain and fever, they are also signs to respond towards the injury or infection. This is an important part of the body's normal healing process. However, this natural response can sometimes lead to excess and chronic production of prostaglandins, which may contribute to several diseases by causing unwanted inflammation. This means that drugs which specifically block cyclooxygenase 2 can be used to treat conditions such as arthritis, heavy menstrual bleeding, and even painful menstrual cramps also. There is also evidence to suggest that these drugs may have a beneficial effect when treating certain types of cancer, including colon and breast cancer. However, it is also very important to know that the use of NSAIDs and other painkillers, it block the pathway of cyclooxygenase enzyme, which reduce the feeling of pain. Now, discoveries are also being made about cyclooxygenases, which suggest that cyclooxygenase 2 is not just responsible for disease, but there are also other functions which are performed by these enzymes. Anti-inflammatory drugs such as aspirin and ibuprofen, they work by blocking the action of the cyclooxygenase enzyme and so reduce prostaglandin level. This is how these drugs work to relieve the symptoms of inflammation and pain and fever. Aspirin also blocks the production of thromboxane and so can be used to prevent unwanted blood clotting in patients with heart disease. And what happens if our body have too low levels of prostaglandins? Manufactured prostaglandins can be used to increase prostaglandin level in the body under certain circumstances. Manufactured means manufactured in industry or artificially manufactured. For example, administration of prostaglandin can induce labor at the end of pregnancy or abortion in case of an unwanted pregnancy. So we can say that if a body has too low levels of prostaglandins, enough labor will not be induced during the childbirth naturally. They can also be used to treat the artificial prostaglandins can also be used to treat stomach ulcers or glaucoma and certain heart diseases, congenital heart diseases in newborn babies. Further advances in understanding how prostaglandins work may perhaps lead to newer treatments for a number of conditions. There are some functions of prostaglandins which we have already studied in last slides. In blood vessels, PGE2 and PGF2, they perform vasodilation and decrease PGE2 decrease blood pressure while PGF2 larger vein contract and little effect on blood pressure. PGI2 also perform vasodilation but led to rapid decrease in blood pressure. And vasoconstriction is performed by TXA thromboxanes A2. For bronchi, it means on lungs and during respiration. PGE2, the role of PGE2 and PGI2 is very important. They perform dilation of the bronchi, inhibit histamine release, and bronchoconstriction is performed by PGF2 and TXA2. While on stomach, we can say that it is observed that decreased acid secretion is performed by eicosanoids. They also increase mucus production, which is performed by PGE2. Decreased acid secretion and mucosal vasodilation is done by PGI2. While in intestine, contraction and longitudinal contraction and relaxation of circular muscles, while increased peristalsis is also and water secretion is also performed by eicosanoids. Similarly, in intestine, spasmogenic increase of fluid and electrolyte secretion is performed by PGF2, while inhibit toxin induced fluid secretion by PGI2. Similarly, TXA2, it can perform weak spasmogenic activity. While on kidneys, natriuresis and decreased level of calcium reabsorption inhibit ATH action, vasodilation, renal release by PGE2 and PGI2, and vasoconstriction by TXA2. So we can say that prostaglandins has wide variety of effects on multiple organs of our body. So they are widespread in our body and are not located on just prostate glands. Then comes thromboxanes. Thromboxanes, the second major class of eicosanoids. They are similar in structure to prostaglandins, with the difference that they have a six-membered oxane ring. They are of several types, for example, TXA1 and TXA2 and TXA3, etc. The most important of all thromboxanes are these types, these three classes, TXA1, A2, and A3. Depending their types they are classified depending upon the number of double bonds in the side chains, as we have already seen in case of prostaglandins. The following structures shows that TXA1 has one double bond in a side chain, TXA2 has two double bonds in the side chain, and TXA3 has three double bonds in the side chain. While leukotrienes. Leukotrienes, they are also derived from 20 carbon polyenoic fatty acids and have a number of substituents. Depending upon the substituents, they are divided into LTA, leukotriene A type, LTB B type, C type, D type, and E type. Each type is subdivided into subgroups depending upon the number of double bonds, which vary from 3 to 5. So we can say that LT, C1, C2, C3, C4 and C5 depending upon the number of double bonds in the side chains. The most important leukotrienes which perform their function they are LTC4, CD4 and LTE4, which has four double bonds. In LTC4, glutathione is attached as a substituent at carbon number 6, while in LTD4, the substituent is cysteine glycine, and in LTE4, the substituent is cysteine. Here, the structures which represent the structure of this diagram represents the structures of LTC4, D4 and LTE4. We can see that apart from main structure of a leukotriene or a eicosanoid, there is just the difference in response to the difference in respect to the substituent which is attached with a sulfur linkage to the eicosanoid in LTC4, LTD4 and LTE4. At LTE4, a cysteine amino derivative is attached. Now we will discuss about synthesis of eicosanoids, how these eicosanoids are formed. The prostaglandins are made up of unsaturated fatty acids that contain a cyclopentane 5-carbon ring and are derived from the 20-carbon straight chain, as we have already seen in their structures. Arachidonic acid is a key component of phospholipids, which are themselves integral component or very important component of cell membrane. In response to many different stimuli, which includes, for example, hormonal stimuli, chemical stimuli, or even some physical agent, a chain of events is set in motion that results in prostaglandin formation and release. These stimuli, either directly or indirectly, they result in the activation of an enzyme which is called phospholipase A. The phospholipase is very important enzyme which start the production of eicosanoids from polyunsaturated fatty acids. Depending on the type of stimulus and the enzymes present, arachidonic acid may diverge down one of the several possible pathways. One enzyme, lipoxygenase, which catalyzes the conversion of arachidonic acid to one of the several possible leukotrienes, which are important mediators of the inflammatory process, while another enzyme, cyclooxygenase, which catalyzes, it means the first one was lipoxygenase and the second one cyclooxygenase. This cyclooxygenase enzyme catalyzes the conversion of arachidonic acid to one of the several possible endoperoxidases. The endoperoxidases undergo further modification to form prostaglandins, prostacyclins, and thromboxanes. So there are basically two pathways of biosynthesis of eicosanoids. The thromboxanes and prostacyclins have important functions in the process of blood coagulation. Prostaglandins E1 and E1 alpha, thromboxane A1, and leukotrienes A3, C3, and D3. They are synthesized from a precursor which has three double bonds, that is at carbon number 8, 11, and 14. So it is known as 8, 11, 14 eicosanoic acid. It is a 20 carbon containing, it's a 20 carbon containing fatty acid which has three double bonds as the nomenclature shows at 8, 11, and 14 positions. While this precursor 8, 11, 14 eicosanoic acid, it is synthesized from linoleic acid. While linoleic acid structure is 18 carbon, it is 18 carbon and has two double bonds at position 9 and 12 as the nomenclature shows. Delta 6 desaturase creates, this is an enzyme desaturase which create desaturation at which creates an unsaturation at carbon number 6 and 7. It creates a double bond between carbon number 6 and 7. The fatty acid 18 alpha linoleic acid, we are talking about alpha linoleic acid precursor, which is a carbon number 18 and has two double bonds at 9 and 12 position and is converted into another precursor which has carbon number 18 with three double bonds 6, 9, and 12, which is gamma linoleic acid. Then chain elongation occurs by addition of two carbon atoms at the carboxyl end. We will see all these steps in reaction form in next slides. Gamma linoleic acid is converted into 8, 11, 14 eicosanoic acid, and here the enzyme cyclooxygenase converts 8, 11, 14 eicosanoic acid into PGG1, the prostaglandin G1. For your ease, the all mechanism, as we have discussed in last 3-4 slides, is diagrammatically represented in this slide. Dietary linoleic acid, which comes mainly from plant oils and has C18 with two double bonds at position 9 and 12. After elongation and desaturation steps, it is converted to arachidonic acid. Arachidonic acid has 20 carbons and is a molecule that has four unsaturated double bonds. This arachidonic acid is further can be converted to important phospholipids and important classes of lipids which are present in our cell membrane. For example, phosphatidylcholine and phosphatidylinositol 4,5 bisphosphate, which depends upon the substitute which are attached at third carbon of the triacylglycerides. In the process of making triacylglycerides, if we start from arachidonic acid is released from membrane lipid. As a stimulus comes at the cell membrane, phosphatidylcholine and phosphatidylinositol bisphosphate, they are converted to their byproducts. For example, phosphatidylcholine with the In the presence of phospholipase A2 and with the help of calcium, it is directly converted to arachidonic acid by releasing its substituent, which is attached at the third carbon of triacylglyceride. While phosphatidyl inositol bisphosphate in the presence of phospholipase C enzyme is converted to 1,2-diacylglycerol, a diacylglyceride lipase, and this diacylglyceride lipase can be further broken down to arachidonic acid or might be further broken down to monoacylglycerol. This monoacylglycerol in the presence of monoacylglycerol lipase enzyme will be broken down to arachidonic acid. So ultimate product of all types of phospholipids is arachidonic acid. So in this way arachidonic acid is achieved from dietary lipids. This diagrammatic reaction shows that linoleic acid in the presence of desaturase enzyme which perform desaturation at 6 and 7 carbon, the product is alpha linoleic acid. Alpha linoleic acid after chain elongation is converted to icosanoid trienoic acid which has 3 double bonds at position 8, 11 and 14. This eicosanoid acid in the presence of cyclooxygenase enzyme which perform oxygenation by taking two oxygen atoms and incorporating into the molecule is converted to first prostaglandin PGG1. This is the cyclooxygenase pathway which shows PG and TX synthesis, that is prostaglandin and thromboxane synthesis. We can see that starting from arachidonic acid has which has four double bond in its side chain. In the presence of cyclooxygenase enzyme, by a cyclooxygenase reaction by taking two oxygens is converted to PGG2, and this PGG2 in the presence of a peroxidase reaction just converted to PGH2. The difference lies just in the substitute which is attached at 14 carbon. An oxygen is incorporated. If an oxygen atom is incorporated in the PGG2, it would be converted into PGGH. We have already seen in last slides that the difference lies among the classes of the prostaglandins just depending upon their substituents which are attached on their side chains. So if we take a summary, if we look upon the summary of the synthesis pathway, we can say that at specific tissues, specific enzymes are produced which perform their reaction and produce appropriate eicosanoid. For example, if PGD synthase is present, it will produce PGD2, prostaglandin D2 type. If PGE synthase is present, it will produce PGE2, and PGE2 might be converted to PGF2 just with the help of an enzyme ketoreductase. And if at any tissue, at any target tissue, PGI synthase is present, it will produce PGI2 from arachidonic acid. And in the presence of TXA synthase, thromboxane A2 will be produced. PGG1 can be converted into PGE1 as well as PGF2. PGE1 is formed from PGG1 by an isomerase, and PGF1 alpha is formed from PGG1 by a reductase. Now this mechanism is shown here in this diagram. PGG1 with the help of an isomerase enzyme, by action of an isomerase enzyme, can be converted to PGE2. If we look upon the substituents which are attached at X and Y position, we can notice that the difference lies just in the ketone and hydroxyl position. It means that a hydrogen atom is attached at the 11th position, that is at the Y position, while PGG1 in the presence of by action of a reductase enzyme which adds two hydrogen atoms at X and Y position, the oxane is converted to an alcohol, so which is a PGF1 alpha prostaglandin. The PGG1 can be also converted to TXA1 and TXB1. Similarly, TXA1 is formed from PGG1 by thromboxane synthetase, and TXB1 is formed from TXA1 by hydrolase. The mechanism can be shown here in this diagram. PGG1, the prostaglandin type G1, by action of thromboxane synthetase enzyme, is converted to a thromboxane type A1. The difference lies just in the substituent and the ring, which is here 1, 2, 3, 4, 5, pentacycline ring. The PGG1 contains cyclopentane ring, while thromboxane contains cyclohexane ring in its substituent along with the side chains. If TXA1 is acted upon by another enzyme, hydrolase, which adds water in the molecule, the cyclohexane ring contains two substituents of alcohol and is converted to TXB1, which is an inactive metabolite. By similar reactions, arachidonic acid can form prostaglandins E2 and F2 alpha, thromboxane E2, and can also be converted to arachidonic acid can directly be converted to leukotrienes A4, B4, C4, D4 and E4. Similarly, 5, 8, 11, 14, 17 eicosapentaenoic acid, that is the eicosanoid which has 5 double bonds, in turn is synthesized from alpha linoleic acid. Alpha linoleic acid, which is an 18 carbon and it has 3 double bonds, is converted into octa-decatetraenoic acid, which has again 4 double bonds and is an 18 carbon by the action of an enzyme desaturase. 2 carbon atoms are added at the carboxyl end to form eicosapentaenoic acid. Finally, a double bond is introduced between carbon atoms 5 and 6 by delta 5 desaturase to form eicosapentaenoic acid. Some eicosapentaenoic acid may be present in diet also. Leukotrienes of series 4 are synthesized from arachidonic acid. Lipoxygenase incorporates 2 oxygen atoms into arachidonic acid in order to make leukotrienes. The product is 5-hydroperoxy-eicosatetraenoic acid, known as HPETE, which is a special class, subclass of eicosanoids. HPETE is dehydrated to LT4A, LTB4, A4B4 is formed from LTA4 by epoxide hydrolase. The mechanism can be shown here. Arachidonic acid, when acted upon by 5-lipoxygenase enzyme and adds an oxygen molecule at 5th position of the long chain and converts arachidonic acid into 5-hydroperoxy-eicosatetraenoic acid. Then this molecule, when acted upon by an enzyme dehydratase, it removes water and a peroxy substituent is added on the long chain, and in the presence of LTA4 epoxide hydrolase, it adds water and LTA4 is converted to LTB4. Similarly, addition of glutathione as substituent to LTA4 converts it into LTC4. That is, if we add glutathione as substituent here in the place of oxygen, this LTA4 will be converted to LTC4. Similarly, alpha-glutamyl transferase enzyme removes a glutamate residue from LTC4 and convert it into LTD4. That is, this LTC4 will be converted to LTD4. Removal of glycine by cystenyl glycine dipeptidase converts LTD4 into LTE4. The reactions can be seen here. LTA4, which is produced in last slide, in the presence of glutathione or LTC4 synthase enzyme, adds a glutathione substitute along with a sulfur to make disulfide bond with the arachidonic acid backbone and is converted to LTC4. This LTC4, with the help of gamma glutamyl transferase, converted to LTD4, and this LTD4 can be converted to LTE4 just by the presence of the appropriate enzyme. This diagram shows the general synthetic pathway of all eicosanoids. When a chemical or mechanical stimuli comes, this stimuli activate the phospholipase A enzyme. This phospholipase A enzyme convert membrane phospholipid into arachidonic acid. Arachidonic acid in turn has two pathways, has two enzymes to act upon, whether cyclooxygenase or lipooxygenase. In the presence of cyclooxygenase, if cyclooxygenase enzyme is present, arachidonic acid will be converted to prostaglandins. Different forms of prostaglandin. First of all, prostaglandin G2, which will be converted to prostaglandin H2. And then this H2 can be converted to prostacyclin, PGI2 in the presence of prostacyclin synthase. There are relevant enzymes and the appropriate products which are produced as the target molecule and the target hormone to act upon the target tissue. Similarly, in the presence of lipooxygenase, HPETE hormone is produced, and this HPETE further in the presence of appropriate enzyme, they are converted to leukotrienes. So the enzyme lipooxygenase convert arachidonic acid towards leukotrienes. This is also a complete diagrammatic representation of the synthesis of eicosanoids, starting from glycerols or phospholipids. In the presence of phospholipase, they are converted to, and then arachidonic acid in the presence of lipooxygenase will be converted to HPETE, and then in the leukotrienes, and different forms of leukotrienes can be made one by one by the action of appropriate enzyme. And while arachidonic acid in the presence of cyclooxygenase enzyme, whether cyclooxygenase 1 or COX2, they are converted to prostaglandins and ultimately thromboxanes. The catabolism of eicosanoids. Eicosanoids are short-lived compounds lasting from seconds to minutes. So they produce their effects locally and are quickly inactivated. Rapid catabolism is essential for termination of their action. They are inactivated by 15-hydroxyprostaglandin dehydrogenase. The prostaglandins are inactivated by 15-hydroxyprostaglandin dehydrogenase. Thromboxane of type A, they are inactivated by hydration to type B. Leukotrienes, that is, when thromboxane TXA1 will be converted to TXB2, automatically the production of TXA1 will be stopped and will be inactivated. The type B will be activated and will perform its appropriate function. Leukotrienes are catabolized by omega oxidation followed by beta oxidation from omega end, just as the beta oxidation of lipids. There are some important functions of eicosanoids. We have already seen their functions in our last slides. Eicosanoids act through cell surface receptors. The receptors are coupled to G proteins. They are short-lived but very rapid and potent. Due to their potency, some eicosanoids are used as drugs. Also, some drugs act by inhibiting the synthesis or actions of eicosanoids. The effects of eicosanoids vary in different tissues. Now, if we talk about actions of prostaglandins, the prostaglandins action is inhibited by lipolysis in adipose tissue. They increase the contractility of uterine muscle and induce labor. They are bronchodilators and hence may be of use in bronchial asthma. PGs, prostaglandins, decrease gastric hydrochloric acid secretion. They cause vasodilation and decrease blood pressure. They increase the permeability of capillaries and ease respiration. They inhibit aggregation of platelets and regulate the blood flow, the amount of blood which has to be stored and which must be flowed from injury. Prostaglandins also stimulate intestinal peristalsis, and regulate digestive system and intestine. They increase the tubular reabsorption of sodium and water, so regulate the salt and water balance. They are required for formation, maturation, and transport of sperms, so help in the reproduction. While actions of thromboxanes, some important functions of thromboxanes are given here. Thromboxanes stimulate aggregation of platelets, so control blood clotting mechanism. While when we will study blood clotting mechanism, we will look upon the functions of thromboxanes in detail there. They cause vasoconstriction and increase blood pressure. Thromboxanes type A2 is a potent platelet activator and vasoconstrictor. Thromboxanes are also implicated in asthma. So during asthma, the painkillers such as NSAIDs, they are banned. They must not be taken. Thromboxanes activate during tissue injury, and inflammation is to reduce vascular content, that is plasma and blood loss. While actions of leukotrienes increase capillary permeability. They are chemotactic agents and attract leukocytes to the site of inflammation. They are powerful bronchoconstrictors during respiration. Leukotrienes have a role in asthmatic and allergic reactions. They slow reacting substance of anaphylaxis is a mixture of LTC4, D4 and E4. While SRSA is responsible for constriction of bronchioles in bronchial asthma. Inhibitors of eicosanoid synthesis. Several drugs produce their effects by inhibiting the synthesis of eicosanoids. Production of eicosanoids begins with the release of a polyunsaturated fatty acid from C2, second carbon of membrane phospholipid. This diagram shows anti-inflammatory drug response towards eicosanoid synthesis. We can clearly see here that anti-inflammatory drugs such as steroids and NSAIDs, they inhibit eicosanoid synthesis. When from membrane lipids, that is phospholipids, they are converted to arachidonic acid by the action of phospholipase A2. And if we take steroids, they inhibit the action of this enzyme, phospholipase A2, by stopping the formation of this enzyme. So ultimately, arachidonic acid will not be formed, which will inhibit the synthesis of leukotrienes and other eicosanoids. Similarly, if we take NSAIDs or other painkillers related to this group, the arachidonic conversion of arachidonic acid towards prostaglandins will be stopped. That is, NSAIDs will act upon by inhibiting the activity of cyclooxygenase enzymes. So prostaglandins and thromboxanes will not be formed. And we have already seen in our last slides during this lecture, the recent lecture, that prostaglandins and thromboxanes, they are related to the pain, to the reflux of pain and fever. So if we take NSAIDs, we will not feel any pain and fever. So they are painkillers. Phospholipase A2 hydrolyzes the fatty acid from C2 of phospholipids. Corticosteroids, they are also inhibitors of phospholipase A2. As we have seen in last slide, they decrease the production of all the eicosanoids. Similarly, aspirin decreases the synthesis of prostaglandin, as aspirin is also an NSAID. So it decreases the synthesis of prostaglandin and thromboxane by irreversible inhibition of cyclooxygenase enzyme. Similarly, phenylbutazone and indomethacin decrease, they are drugs, they decrease PG and TX synthesis by reversible inhibition of cyclooxygenase. Some leukotriene receptor antagonists are used as drugs to treat allergy and asthma. Receptor antagonists. Some leukotriene receptor antagonists. They are used as drugs to treat allergy and asthma. Such drugs include montelukast and zafirlukast. Thank you.